So hi
Go ahead.
everyone. Um, I'd like to talk about today's the structure and dynamics in the model of the macaque visual cortex. So the macaque visual cortex is very much like our own. And on the picture on the right here, you see a picture of the side view of a human brain. So as information enters the eye, going to the retina, and in one synapse, it goes to what's something called a lateral geniculate nucleus, shaped like a knee. And in another synapse, it goes to the back of the head, where the visual cortex is located. So the model that I like to talk about goes from LGN. I skipped the retina part. It goes from LGN to the input layers of the visual cortex. And I will be discussing what is in the magnostream, one of the two main visual pathways into the visual system. Now, this is what I'm going to describe is what's considered to be realistic computational modeling. Now, no model is completely realistic, or it wouldn't be a model at all. So by realistic, what I mean is realistic in the world of models. And this is joint work with Bob Shapley, who's a neuroscientist, experimentalist, and Logan Charaka, a younger fellow. Many of the simulations that you see are done by him. So let me start by giving you some idea of the structure of the network that I'm discussing. On the left, this is not part of the network. This is intended to symbolize the external stimulus that I'm going to present to the network. So the network has three components: the LGN, okay, the lateral geniculate nucleus, and layer 4C alpha, which is the input layer to V1, the primary visual cortex. The input layer interacts with another layer of V1, layer 6, and they talk back and forth. Layer 6 actually also gets some LGN input, but the main input layer is 4C alpha. So now both LGN and 4C alpha are modeled fairly realistically, meaning that we follow neuroanatomy in every way that we can as far as the network neurons and structure goes. Layer 6 is very complicated. It has a lot of very large variety of neuron types, and also interacts with other parts of the V1. So we're not able to model that realistically. But in any case, the main object of interest are these two parts, LGN and 4C alpha. Now, I cannot really leave out layer 6 because layer 4C alpha gets gets feedback from it, so it has to be there. Okay. So what I'm going to discuss are the structure of LGN and layer 4C alpha, and also the dynamics of neurons in both of these. And in particular, in 4C alpha, I'm going to discuss it on three different levels: on the level of individual neurons, on the level of local populations, and also across the cortical surface. Okay, so structure and dynamics of LGN. LGN is a fairly simple object, in the sense, not simple at all, but simple compared to cortex, in the sense that, well, there are 6 different layers. Here's one layer. Two things that you need to know about it is that one is that it's very sparse. Now, just to shock you a little bit, if you stare at the moon, in the area that covers about a quarter the size of the moon, there are only 10 LGN neurons, on the order of 10 LGN neurons on the magnosheets. So that's very few neurons covering a very large part of the visual area. And the second thing you need to know about it is that there are 2 types of cells, on and off, and here's how they function. So LGNs basically function as linear filters. That's why their dynamics are relatively simple. So the input current reflects fairly well what's in the output. So what goes into the input current in LGN, first there's a background current that's always there, no matter what you do, it's there. Okay, and then it's plus or minus some, depending on whether you're an on cell or an off cell, another quantity, which is it's an integral of the light intensity map. So L of X is this light intensity map that describes what the external stimulus that comes in. And there's a spatial kernel, A, and there's a temporal kernel. So the spatial kernel, this is a 2-dimensional object. LGN is really a sheet, a 2-dimensional sheet. So this is the radius of the spatial kernel. So basically it just sees a little spot with a little bit of a center surround effect, but it's fairly weak. This temporal kernel is more interesting. It has a shape like this, which is not the usual shape of a kernel. But what that means is that LGN is a change detector. When you integrate it against something like a sine function, what you're doing is taking a derivative of the input signal that's coming in. So for on cells are excited when it goes from dark to light. So each LGN neuron only sees a little tiny little spot in visual space. So it focuses on the luminous of that spot. And when it goes from dark to light, this derivative tells the on LGN cells to spike. They get excited. And the off cells do the opposite. They get excited when it goes from light to dark. And that's basically all that there is to know for the purposes of this talk about LGN. Okay, so again, it's where LGN has, so something I did not stress, I should say, is that LGN has a retinotopic map. Now there's a map of the visual field on our retina, everybody knows that, but that map is passed to LGN and actually onto cortex. So in other words, LGN has like, there are spatial locations in the sheet will reflect the spatial locations in the real world. But so that's something that LGN has. But on top of that, it really just does this integrating and taking derivatives. There is certainly, it does not recognize edges. So there's no such thing as orientation selectivity or anything like that. It's just very, very simple. Two kinds of cells. One goes on when it goes from, when things turn from light to dark, and the other one goes on when it goes from dark to light. And that's all the dynamics and structure of LGN. Now, going to the structure of the model cortex. Now, cortex, so I'm mostly talking about input layers of V1. And one of the most salient features of V1 is orientation selectivity, in addition to having this retinotopic map. What orientation selectivity means is that each neuron has a favorite orientation. So it also has a tiny receptive field. And if it sees an edge aligned with that or its favorite orientation, then it's really excited. And when it's orthogonal to it, it doesn't respond at all. And in between, it responds by variable amounts. That's what's meant by orientation selectivity. Now, this is a picture of a real piece of V1. It shows that the neurons preferring the same orientation tend to be grouped together. And these are called orientation domains. So you will see the neurons preferring vertical, colored red in this imaging map, and neurons preferring like horizontal are colored in this blue-green. So you see that they are grouped together. Okay, so that's a real piece of V1 cortex, and here's the model. Okay, so then the model, this is a 9 hypercolumn model. Each square, hypercolumn, each square represents a hypercolumn. There's no such thing, you don't see hypercolumns here. Hypercolumns are just structures that we made up to make it easier for us to talk to groups of orientation domains. So each hypercolumn is further divided into different, these are the regions that correspond to the colored regions. Okay, they are intended, they are regions of intended orientation with intended orientation preferences. So just to give you some idea of the model, which follows real anatomy, each hypercolumn is, this is at about 5 degrees eccentricity, fairly close to when you look directly in front of you. So each hypercolumn it represents about 1/4 of a degree by 1/4 of a degree, like roughly the size of 1/4 of the moon, okay, when you look up at the sky. And in this layer alone, there are 4,000 neurons in each hypercolumn, 3,000 excitatory and 1,000 inhibitory. That's just one hypercolumn, one layer. So you can see that these 3,000 and 1,000 here, as opposed to LGN, which has only about 10. It's a very, very big difference in number. And so in this model, the neurons are modeled by leaky, integrating, and firing neurons. And here's the connectivity. So I want to stress that to the degree that we can, everything we try to do is models realistic densities in real cortex and realistic length scales. But of course, hypercolumns are not square, that we made up, so that otherwise it's very, very hard to communicate with the computer. Okay? So, so this is an E neuron, and you're looking at randomly drawn, pretty much isotropic groups of neurons that are connected to it at connection densities that are that mimic real cortex, and it's quite sparse, as you can see. Whereas this is E to I, so an I neuron sitting in the middle, and E neurons like that. And on the side, most of the neurons in this layer are basket cells. They have very short axons. That's why it's the cloud is smaller. So this is mostly the structure of the layer 4C alpha. Layer 6 is a layer that's very similar to this, but let me focus on 4C alpha. So this is pretty much the structure of 4C alpha. So now the next thing I have to talk about is how is LGN connected to 4C alpha. So in particular, I need to answer the question, how is orientation selectivity conferred on V1 neurons? As I said before, LGN has no orientation selectivity. It only goes light, dark, light, dark. It only cares about changes in luminance. So there is no way you can see edges from they don't know anything about edges at all. Whereas V1 neurons, they all have favored orientations. And yet there is nothing between LGN and V1. So how did this orientation selectivity come about? Okay. Well, the answer was suggested to us or provided for us, which however you see it, by Hubo and Wiesel. So in this scheme, cortex is divided into regions of intended orientation preference, as I've shown you before. So these are the bars indicate the intended orientation preference. And the task in building the model is to each neuron in V1, we're going to assign a handful of LGN cells, a number of LGN cells that are supposed to be spatially aligned in 2 or 3 stripes of on and off. This is what Hubo and Wiesel showed us, and I'm going to show you what I mean by that. But first, a word about the number. Up until up until fairly recently, people used to think that the number of LGN cells connected to a single V1 neurons, it's like in the dozens maybe, and it was only found not so long ago, mostly from the experimental result of Angelucci and also the sparseness of LGN cells also, that is really only very few, it's only a handful. Okay, so here's what I mean by rows of on and off. So these unfilled and filled circles represent on and off LGN cells, and this is a row of on and this is a row of off. So Hubo and Wiesel says that we get cortex gets orientation selectivity by connecting to these rows of on and off cells. So now the moment that we start to do this, there's a combinatorial geometric problem that comes up, is that, well, since you can't reach very far, so like roughly this many, how many on-off stripes can one draw from a set of about 15 to 20 LGN cells, half on, half off? You can see that it's really not so easy to draw. I can draw a horizontal one easily, but there are not that many angles that you can draw from. It's very, very discrete. And how do you represent orientations around the clock? And there's another issue, is that the distance between the on and off have to respect certain laws about that show up in the spatial frequency preference. Okay, so the answer is it can be done, but barely. And here are some example stripes that we have. So for example, here is that this is a stripe, an on stripe, an off stripe, and an on stripe. So this is a 3-stripe cell with 3 of the stripes according to Hubo and Wiesel. Here's one with 2, and off, and then on, and so on. So maybe I should say a word about how why are these stripes? How do they confer orientation? So take an example of this 2 off, 2 on one. If you have a grading that's vertically aligned, I am claiming that a V1 cell that's connected to these 4 LGN cells will be very sensitive to vertical edges. And that's because when a vertical grading comes, imagine it's coming from right to left like this. Oops. Okay, it's coming from right to left like this. If the spatial frequency of the grading matches this distance between the on and off row, there will come a time when the on cells are very happy because they see white, and the off cells are very happy. So all 4 cells are very happy and they spike together. And when all of the LGN cells spike together, it's very powerful. It sends a very, very strong signal to cortex. Now imagine that you have a grading that's aligned some other way, coming say from some angle like this, okay, so some different angle. You will excite 1 or 2, maybe 3, usually 1 or 2 at a time. You will excite them eventually, but they are not excited simultaneously. And so the message sent to cortex is much weaker. And this is what Hubo and Wiesel suggested, and we verified in the model. Okay, so this is all that I'm going to tell you about the structure, except that I haven't said anything about layer 6. Layer 6 is the firing rate in the model is indexed to layer 4. So layer 6 is another sheet. Think of it as sitting below layer 4. So it has the same kind of spatial locations. And so what we do is to just make layer 6. Layer 6 is very hard to model because it's very, very complicated. So all that we do is to have it feedback something which is indexed to layer 4. If layer 4 firing is high, it sends back a feedback that's high, and layer 4 slow is low in the same location at the same time. Okay. So this completes the description of the network. And next I'm going to talk about dynamics. But before I get into that, I want to say that once the parameters are chosen, I'm not allowed, the modeler, in other words, me, I'm not allowed to touch the model. The model is going to, so once this input goes in, the model will compute on its own. LGN integrates all these things, sends to V1. The neurons here interact with layer 6, and layer 6 comes back like that. The model is going to compute on its own, and it's going to follow the mechanics of neuronal interaction in the real cortex as far as we know. Okay. So about dynamics, first I want to talk about the dynamics of individual neurons versus local populations. So, and both of these things have been shown to, so every step of the way in the building of this model, we have tried very hard to make sure that everything matches data. So the number of sets of data that are matched, I don't really know. It's many dozens. It's hard to count whether they're the same or different. Okay. So first, for individual neurons, there's something very, very well known, that if you take an individual neuron and you measure the time intervals that passes between spikes, you will see a distribution that looks like this into spike intervals. It looks very much like an exponential distribution. It need not be exponential, but it just has a very long tail. But if it was an exponential distribution, it would really basically mean that the next spike is going to come at a fairly random times. It's like a Poisson process, right? And this is when it's driven by a grading at 4 Hertz. So you can see that roughly 250 milliseconds later, there's another little bump. But in short, if you look at a single neuron, the spike firing times of a single neuron appear to be very, very random. In fact, one way that one could rationalize that is that each neuron receives a fluctuating current because it receives a current from many other neurons. So the sum of which is a fluctuating object function that produces a random walk-like behavior with a drift up towards threshold when you are driving it. And so ISI can be modeled as a first passage time from reset to threshold as inverse Gaussians, and they have shapes that look like this. Now, individually, this is what neurons do, but when you put them all together, something else happens. Okay. In the model, they produce a gamma rhythm, which I'm sure most of you know about. So this is a raster in the time in the horizontal axis, and neuron number. Blue is I neuron, red is excitatory neuron. The ones that fired further down below, they fire more because they're complex cells. Okay, so you can see that the spike firing is very far from homogeneous. It produces a rhythm, and I want to emphasize that no organization, there was nothing that I did to cause this to happen. The neurons are certainly not grouped in any way. They are kind of randomly picked, and yet they self-organize into this kind of spiking pattern with the frequency at a gamma band. Okay, and this is an entirely emergent behavior. Okay, so just to say a little bit, so gamma band rhythm is detected, as you all know, in many parts of the brain, and it also shows up in models. You take a group of neurons, you randomly connect them together, and a gamma rhythm will, if you respect the physical time constants, I should say, and respect the fact that E and I are in good balance, then a rhythm will appear, and it's almost hard to get rid of. It's just there. Okay, so this is just a picture from the previous slide. In case you think that these are population spikes, here's the percentage of neurons that are firing at any one point in time. As you can see, most of the time it's less than 10%. Occasionally you get a big one like 20, but is extremely far from synchronous firing. Okay? And if you look carefully, it's actually not the same ones that form that group together to fire also. So neuroscientists have done a lot of computation of the power spectral density. So one of the reasons for looking at this is that you look at this, they call this a rhythm, but it's not really exactly a periodic, is it? Some are really wide, some are narrow and get messed up, and some are kind of come on stronger, and some are fainter. Okay, so this is described by this power spectral density. And what this is that n is the total number of neurons. So you look at the time and divide it into a lot of different time bins, and then you look at UN is the instantaneous firing rate, the fraction of neurons that's firing, rather not instantaneous firing rate, sorry, is the fraction of neurons that's firing in the nth bin. And so what you get is really a signal, an infinitely long signal, which is very far from periodic, but you pretend that it is, and you look at the first like 200 milliseconds or so, and you compute its Fourier coefficients. And then you time shift that time interval by a little bit, and here's what you get. So this is a fairly realistic picture in the sense that you almost cannot tell it apart from the real experimental data. So this is time. So here's very long. This is 1 second. Between here and here is already 1 second. This is the frequency. And you can see that the power is mostly concentrated around like here, maybe about 60 hertz, which is in the gamma domain. But it wanders. So this is the wandering frequencies. This is a character of gamma signal, and it's completely, entirely emergent behavior. It's 60 for a while. It could disappear. It could drop to 50. It sometimes goes up to 80, 90. It just wanders around. That's a caricature of it. Okay, and so there are different recurrent excitation inhibition is what is it's an explanation for it. Since I'm running short in time, I'm not going to explain that, as it takes a little bit of time to explain. Okay, so what I'd like to show you are some pictures of some. This is what the model cortex looks like when it's stimulated. So look at first this picture. This is when you're passing a vertical grating through. Now, here's the key, the translation. If you go to the model cortex, these are the regions that are colored purple, are the regions that are supposed to be intended to be sensitive to vertical grating. They are wired, meaning they are wired as such. Of course, you don't wire everything perfectly, but they are predominantly wired to prefer vertical. And indeed, when you present a vertical grating, these are the parts that light up. Lighting up means that it's firing high. This means dark. This is E and this is I, and you can see that the eye neurons are more broadly tuned. So it's got a much bigger footprint in this picture. So if you go to horizontal, 90 degree, you will see that these are the diamonds, and they correspond to these. And what if I go to 45 degree? This is a 45 degree grating. If you look here, you don't see 45 degrees, but you see 30 and 60 degrees. So what a 45 degree one responds to is some combination of 30 and 60 degrees, so you can see the correspondence between this intended wiring and the actual response of the neuron. So just to focus on the central hypercolumn, people talk about this pinwheel structure. You will see as you go along 0, 25, 45, like that, okay? And to remind you again, this is the connectivity. It does not respect this, it crosses the orientation boundaries. It is fairly isotropic. Okay, so this is I have a movie that I like to show you, and this is exactly what you saw before, except that what I showed you earlier was average over 1 second, and this is happening in real time. Well, not in real time, I've slowed it down or you wouldn't be able to see it. So here is the time clock in milliseconds. It's gonna be, it's moving. And you can see that I'm starting with a vertical grating, and these are the diamonds that correspond to the vertical grating. So I want to emphasize that the model is really computing on its own, and it's following the way the brain computes. In other words, it's alive, okay? And so here it goes. Okay, so this is a vertical grating going. In a moment, it's going to change to another grating, change to a 25 and 22.5 degree grating. So you notice that a different part of the cortex is lit up. Okay, in a moment, it's going to change again to a 45. This is This is the horizontal grading. Okay. Okay. So so much for the movie and a summary and conclusion. Okay. So we have built a structurally realistic model of the macaque V1 cortex, and by structurally realistic, I mean following the anatomy of the real cortex on the level of neurons and circuits, because we did not model what goes on inside neurons or intracellular properties are not modeled, but on the level of neurons and circuits is as realistic as we could do it. And with biological rules of dynamical interaction, meaning that LGN are change detectors, we model them as such. The LGN to V1 connection, we follow Humo and Wiesel. The dynamical interaction, the length of axons, and so on are intended to model real cortex. And what we found was that in cortical dynamics was that it's really quite different if you look locally versus looking globally. If you look locally, it mostly depends on an EI balance. You see if there's a balance of the excitatory and inhibitory neurons, as well as the push and pull that produces the gamma rhythm. But as you look across the cortical surface, then it's a different story. It depends on many, many things. First of all, it follows the stimulus. It better follow the stimulus or we wouldn't be seeing anything. But it's also a complicated balance between the feed-forward input, the lateral contribution, and feedback. And here, both structure and dynamics of feed-forward, lateral, and feedback all count. So the feed-forward would have to be what LGN gives it. The lateral in this layer, there are no long-range connections, but otherwise it matters whether or not there are long-range connections, and it matters how much feedback. Mathematically, this can be described as a non-equilibrium steady state. It's a concept borrowed from statistical physics, and there's a fair amount of mathematics and physics associated with that. So this is all that I wanted to say. And my team, again, is Logan Charaka, the fellow on the left, Bob Shapley, neuroscientist, and myself, and thank you to the Schwartz Foundation and to NSF. And thanks for listening.
Thanks, Lai-Sang. So if there are questions, please type them in the Q&A box. In the meantime, I had a question. So what happens if you present, so this is kind of a two-part question. What happens if you present more complex stimuli? And how could you, and as a result, to turn this around, how would you verify this model accurately captures cortical responses?
Okay, so the structure of this model is certainly not built to only replicate the dynamics in response to gradings. Okay, well, but it's very small. It only has 9 hypercolumns, which means that you cannot. It only sees an area that's roughly 0.75 times 0.75 degrees. So first of all, I will have to extend it spatially in order to see more interesting objects. But as far as the structures go, this is all there is, basically. So if this model is supposed to capture everything in this tiny piece of the visual cortex, independent of the nature of the stimulus.
And so what would happen but it excludes top-down projections, right? So that could modulate those responses.
That's right. If you in trying to model the visual cortex, I think that we have a fairly good chance of success modeling the volley of information that's coming in. Top-down things are very hard to model. So, for example, V1 talks to V2 to MT, to all these things, and MT and these other V4, V5, these areas, they talk to other regions of the brain, and it bounces back. We're not in a position to model, certainly not a detailed, accurate modeling of this kind to deal with any of those. So what we are really trying to capture here is the input volley of information. So if you've thought about it and decided, well, you know, yes, it modulates what you see, but still it's dominated by the input information in these layers.
Thank you. And so this is all the time we have. So thank you to Lai-Sang and to all the other speakers. As I mentioned before, We have a Slack channel devoted to this particular session, parallel session. I just checked that there's the most of the questions that were posed and not answered directly during this time have been answered on the Slack channel. If you have further questions, this is also a good place to ask the speakers again. So thanks again. And I think we will reconvene in about 25 minutes, or rather there's going to be another talk in 25 minutes. And that's it.